Good evening. My name is Dr. Ross Boucek. I'm with Bonefish and Tarpon Trust, and thank you for tuning in to our second live stream event to provide updates on the ongoing fish spinning phenomenon and sawfish mass mortality event occurring in the Keys. In our previous update, we summarized the discovery by Mike Parsons and his team of highly elevated concentrations of toxic algae species that live on the bottom that are co-occurring in places where we're seeing consistent and intense fish spinning behavior. In addition to that, results from Dr. Alison Robertson on the toxicology side identified that consistent toxins are both found in the fish, in the water, and in these algae samples that we've been taking in the lower Keys. This connection between the algae, the water, and the fish is our best lead to date, and one we've been pursuing with research for the last 2 months. To start from the beginning, we first detected this by our best approximation becoming an event in the beginning of October. In January, a interinstitutional collaboration began between LKGA, Bonefish Tarpon Trust, Florida Fish and Wildlife Conservation, Florida Department of Environmental Protection, and several academic institutions, including FGCU, Florida Gulf Coast University, University of South Alabama, Florida International University, Woods Hole Oceanographic Institute, Bimini Sharklab, and also Florida State University. This involves over 50 researchers dedicating significant time to investigating what is causing this, what are the impacts to our ecosystems, and importantly, what can we do about it? In addition to this, over 70 different fishing guides, both from the inshore and offshore fleets from across the Keys, have committed time to collecting water samples, fish samples, and also sharing their knowledge of what's going on real time to help inform where we look for things, what we look for, and a better, more focused understanding of the system and the research we do. Together as a working group, we've explored 4 lines of inquiry. Is this a human contaminant issue? Is this something coming out of our sewer system, off our roads, or off construction sites? So it could be leading into these behaviors that we're seeing in fish and the deaths of sawfish. As I mentioned before, teams have investigated whether or not this is some kind of toxic algae that lives off the bottom that's producing toxins that's affecting our fish. Or is this something in the water column, like a red tide that's biologically produced, also causing these neurological effects? And last, is this a fish health issue, a virus, parasite, or chronic malnutrition that's manifested itself in the behaviors that we've seen today? To date, we've documented that this is indiscriminate across multiple species. Basically, everything that swims in the water with gills has been affected to some degree, the exception being bonefish, tarpon, and barracudas seem to be less affected than others. The most affected species seem to be the jacks, the permits, the jack crevalles, the yellow jacks, and all those species in that group, in addition to the skates, the sawfishes, and the stingrays. Inquiries along those 4 themes of work, whether that be human contaminants, toxic algae on the bottom, and etc., have shown that for human contaminants, no common chemicals have been found in the water that are at detectable limits or above background levels in any of the places where spinning fish have been observed. Red tide samples have shown no evidence of anything in the water that would be biological and harmful and as an algae. And human health or fish health metrics have shown that these fish are otherwise healthy except for the fact that they're spinning. But the lead that we mentioned earlier of the discovery of the harmful algae living on the bottom is what we've been pursuing to date. The people on the call today are myself, Dr. Ross Boucek from BTT. We have Dr. Mike Parsons from Florida Gulf Coast University. He is a harmful algae bloom ecologist, tries to understand when, where, and why these things occur and what their impacts are to the ecosystems. We have Dr. Alison Robertson from South Alabama, who is an ecotoxicologist evaluating the effects of human contaminants and toxic species, biologically produced toxins on fish, wildlife, and people. Last, we have Dr. Matt Smukall from Bimini Biological Field Station. He's been studying the communities of sharks in the lower Keys for a couple years now, with specifically his research really is focused around Cudjoe Key, which was ground zero of this event. And it's very informative information based off the work that he's done prior to this event and leading up to it. In the rest of the live stream, we'll talk about updates from Mike Parsons and what his team is finding with the algae, new toxicology results or updates on those, and of course, what's been going on with the shark species in the lower keys that could help us better understand this issue, particularly as it applies to the elasmobranchs. So with that short introduction, I'll move over to Mike Parsons and let him provide us an update of what he's been doing and what he's been finding down here in the keys related to fish spinning.
Great, thanks Ross, and thanks to the Bonefish and Tarpon Trust for this opportunity to talk about some of our findings. So we've had 3 collection trips so far in the keys. We've collected probably 300 or 400 samples, looking at different kinds of microscopic algae. These are single-celled photosynthetic microbes that they're about the size of a grain of sand, so they're pretty small. The ones we're interested in tend to live on the bottom, and they tend to live on seagrasses, on seagrass blades, or on macroalgae or seaweeds. Sometimes they'll get up into the water column, but they prefer to live down associated with these seagrasses and seaweeds. And so in collecting these samples, we've also collected water and fish samples for Al, so she'll talk more about that. But in concentrating on our algal samples, on our seaweed and seagrass, what we're noticing is that we are seeing elevated concentrations of one genus of these microalgae in particular, Gambier discus. And so Gambier discus is a dinoflagellate. So that's a single-celled microscopic algae related to Florida red tide, Karenia brevis, but not that closely related. It'd be like comparing a horse to a tiger, if you will. So they're both mammals, but they're different. So Karenia and Gambier discus are both dinoflagellates, but they're not that closely related. So when we look for these Gambier discus cells, we try to see, do they prefer to live in some areas? Do they prefer to live on seagrasses or on some seaweeds versus others? And during this event, since February, really, February and March samples, we see that Gambier discus seems to be really associating with turtle grass, Thalassia. And we do see it on some of the seaweeds, but it's not really elevated like we see it on the seagrass. And also we're seeing higher levels of Gambier discus on the seagrass around Cudjoe Key and in the lower key sites that we've been sampling. It's pretty much at normal concentrations when we look in the middle keys, around Long Key, Marathon, when we look out on the reef and in the patch reef sites, other bay sites or Gulf sites. So it seems to be, like Ross said earlier, a ground zero right around Cudjoe Key and Big Pine Key. And so one thing that we're moving towards right now is trying to understand this difference. Why do we have this hot spot here? Why aren't we seeing this everywhere, this elevation? And why are we seeing this association with Thalassia in particular? We have more samples to look at, so this might not be something that really pans out in the future. But again, it's one of these leads that we're pursuing since that's where the data are leading us. So I think that's it for an update on the algae, Ross.
So you sampled, yeah. So in March you sampled across the keys, right? And
yeah.
15 different places that you've been sampling for 14 years. Did you find anything else that was out of whack? Any other these?
No, for the most part, you know, when we compare it to our long-term data, all these other sites seem to be normal. You know, the cell concentrations are the same. Al has a lot of samples to look at the toxin data, so she'll be working through those samples to see if there's anything different there, or if it looks like it's, you know, normal quote unquote conditions. But yeah, we're not really seeing anything different at these other sites, so that makes it a little puzzling.
Yeah, indeed. Do you think it's related? Is there a connection between the heat wave potentially, or do you have any at least speculations of what is causing this or new leads on that?
Yeah, with I mean, the heat wave is such a significant event that happened last summer and we saw a lot of reef impacts, a lot of bleaching, cyanobacteria growing on the corals. So we know it's had some very large impacts. And it's unclear if we see it in the seagrass beds and the meadows and the flats as much. So we're trying to see what data are available. Like Matt has data before this event with the shark research and his team have been doing. So we're trying to gather data around the lower keys where we have not been regularly sampling. We tend to be in the upper and middle keys more, and just try to see, is there any evidence that the summer maybe, that hot summer maybe kicked off something that ended up, producing and showing itself into the fall and the winter months. So that's part of the mystery that we're involved in right now, for sure.
All right. Well, I'm sure we'll come back to you, but yeah, we're looking forward to having you back down to sample some new locations or continue sampling your previous locations here in June.
Yeah, yep, we'll be down there in a couple of weeks.
Cool. So Dr. Robertson, your focus is on the toxicology, work closely with Mike. Can you provide any updates on what's going on with you? New leads, causes, anything?
Yeah, for sure. Thanks. So we're with the University of South Alabama and the Dolphin Island Sea Lab. That matters because we're working in both locations and require all of the facilities at both places at the moment to undertake this massive task. So what we've been doing from all of the samples that's been collected, both through Mike's team at FGCU, but also through the Florida Fish and Wildlife Commission scientists and yourself and others at BTT. We've been receiving water samples. So we've received several hundred water samples since the beginning of this, since the beginning of our involvement, which has really been since about February. And in addition to that, we have about 150 or so algal samples. So these are benthic algal samples that were collected from these seagrass and seaweeds that are on the bottom. And hundreds and hundreds of fish samples, representing about 30 species or so. So of all of that, why does all of this matter? What are we testing for? We've been testing for a lot of the benthic algal toxins. So when Mike noticed that there were elevated levels of these benthic algae or dinoflagellates in the water column, that led us to target at least some of those known neurotoxins. And so they include some that we know of as ciguatoxins. They have been associated with a syndrome called ciguatera poisoning. And that's something that's well known in tropical and subtropical regions. So it's not so unusual necessarily to find those toxins in fish. And we have been finding levels of those toxins in a number of fish. But those seem to be slightly elevated and more prevalent in fish that have been symptomatic. So that's been one of our leads, but not the only lead. In the algal samples that we've been working on, we've detected a number of other classes of toxins, and they have all sorts of long and crazy names, which we don't necessarily have to go into, but all of them are known to be neurotoxins affecting various targets. What we found through our experimental work was that the combined effect of these toxins seemed to replicate these spinning behaviors. So that's important, validating that we might be on the right track, but we're also looking for any other components, toxins or otherwise, that could be stimulating these sorts of behaviors, because they are the sorts of behaviors that we do expect from any compounds that can cause any kind of neurological impact. So we're also looking at these samples in through some methods that are non-targeted, meaning we're not chasing after one specific thing. We're keeping our eyes open. And that means that any one sample might generate actually hundreds of different subsamples as we fractionate or separate different components based on their chemistry. In doing that, we can start to identify things that have the biological activity, even if we don't know what it is. And that can be really important as we try and solve some of this mystery. So we have been finding a number of the benthic algal toxins, though, across fish, the algae, and in the water samples. Those seem to be the highest and most prevalent in the areas in the lower Keys, although we are seeing some levels in other locations as well. So that just requires more investigation and more sampling at this stage.
Well, thank you. Do you have any idea of like exposure pathways or anything along those lines, or?
Yeah, definitely. So what seems to be happening is we're looking not just at fish tissue, like the edible tissue that you might eat. We're also looking across other tissues of the animal, and that might be in their blood, in their gills, liver, kidneys, and so on. And we found elevated levels in when toxins are present in the liver, which suggests recent exposure, because the liver is kind of that central station to the body where contaminants are going to go and get rerouted and redistributed and try to be eliminated from the body. But also findings in the gills. So the gills have been one of the tissues where we found the most significant bioactivity. And this led to the finding of a new toxin class, at least from our perspective, had not been seen before in the Keys. And that correlated to our prior findings of that same toxin in some of those benthic algal samples. So these are important findings, and we're trying to see if it matters, right? So we think it does because the gills have been consistently a tissue that in our bioassays results in loss of neurons, because we do these tests with neuronal cell lines where we can test various extracts, and consistently in these fish that have been affected, we see that the entire cell population is wiped out. That doesn't mean that it's a huge risk necessarily to human health. Of course, this is gill tissue, and that's not something that's commonly consumed, but it is really important and suggests a waterborne exposure of these toxins. So.
Well, that's good news, I guess, for waterborne exposure versus the other way. Would you be, if it's waterborne exposure, shouldn't eat the gills of fish, I guess? Is that the take home, or?
Yeah, don't floss your teeth with the gill rakers, maybe. But at the same time, a lot more work would need to be done to determine the risk of those toxins that we've identified in the gills. But in the muscle tissue, so in the tissues that we would potentially eat, we have seen a lot of these benthic algal toxins present and bioaccumulated in those tissues, but they are below those guidance levels set by the US FDA and others for safe consumption. So at the moment, at least in the samples we have been analyzing, it doesn't seem to be a huge risk for human health. But we do always, right, like follow fish guidance advisories and swimming advisories and all of those things. But if, I think we've all said this before, if fish are actually dying around you, probably don't eat those fish or swim with them. But at the same time, the toxin levels in the symptomatic fish that we've seen don't appear to be above any kind of human health guidance level for those natural toxins.
That's good news. I guess anything else new and exciting on the horizon that we're working on?
Yeah, we're working on a lot of experiments in fish to look further at the effects of what might be in the water. So these are experiments we're doing in conjunction with Mike at FGCU. So we want to collect a lot of water, and by a lot of water, we're talking hundreds of gallons of water, and to expose fish in that to that water directly, but then to take everything that's in that water and parcel that down into a much a very, very concentrated kind of sample. And we want to look at that. So if we can then see direct exposure in fish, we might be able to characterize, have the same pool of water that was causing that effect directly. Hopefully that makes sense, but that requires a lot of water and a lot of work. It's not as simple as it sounds to just concentrate thousands of liters of water. But we're going to give it a go. And we're also doing direct toxin testing where we're looking at the combined exposure of natural toxins. These, however, are not toxins that you can buy off the shelf. This is not something that we can go down to the local store and get, or the scientific companies, have only minuscule amounts, if any at all. So we have to purify a lot of these toxins ourselves from the environmental samples we're collecting in the lower Keys. So with that material, though, we're then doing direct fish bioassays where we're testing to look at behavioral change and then bioaccumulation and see if those all match up to what we're seeing in the field.
Awesome. Awesome. Yeah, the experiment sounds pretty elegantly simple. Get some weird water and throw some healthy fish in it and see what happens. So
yeah, exactly.
Excited for the results of that.
Mm-hmm.
Well, thank you, Dr. Smukall, the shark expert. Tell us about sharks. What have you been doing? What have you been seeing in the water? And what does it mean for the sharks?
All right, hardly an expert, but thanks for having me join. So yeah, I'm with the Bimini Sharklab. So we have a long history of doing shark research in the Bahamas and then also through South Florida. And actually, for the past 2 years, so going back to 2022, we've been doing research in the lower Keys, so around Big Pine, Cudjoe, Sugarloaf Keys, those types of areas. And it was actually because a lot of the sharks from the Bahamas had moved to these areas, so it was a natural spot. We wanted to study a little bit more extensively based on some of our previous research. And so that actually was fortuitous in that when an event like this does happen, we have surveys that actually go back. One of the hardest parts when these types of things happen is that we tend to only get funding or research or something after the fact. And so having the ability that we actually have information going back to 2022 on catch rates from our surveys, or even just on biological samples from some of the sharks that we had caught previous to whether it was the heat wave in 23 or then the subsequent spinning behaviors over the winter. We've been looking into some of those potential things. As far as with running toxins and things on the blood samples, we're a bit we're kind of being patiently waiting for other people to be able to follow their leads and hypothesis to get a better idea of what we might want to start screening some of those, whether it's muscle samples or blood samples for, just based on the fact that we have the blood samples, but they run out very quickly if you start going down too many different paths with it. A lot of our samples were originally collected for things like reproductive hormones or potentially trophic ecology studies. And so we have to be careful with where we're going. And so we're hoping as some other, whether it's captive experiments or things like that, get a better idea of what these toxins, the most likely ones are, then we can start running those with some of the shark samples. What we do have now to date is the catch rate data, which is basically, how many sharks are we catching per number of hours that our hooks might be in the water and things like that. And so what we noticed is that so in our surveys in February and April of 24, so a couple months ago, when this was really starting, especially in February, when this was really starting to hit the peak of it, as far as with the sawfish mortalities and stuff, we were looking at the shark aspect of it. And we were noticing that areas that the year previous, so the same month, but the year previous in 23, we had a very healthy number of sharks that we were catching per survey. And then going back to those same areas in 24, we were sometimes not catching any sharks at all on our line. So for an example, we would go to certain spots and, on what we call one of our surveys, we might have 5 to 7 sharks out of 25 hooks that are in the water. And then you go to 24 in the same exact area, same time of year, roughly the same water temperature, things like that. And we would have 0 or 1. And so, you definitely need more data to make any sort of like hard and fast, kind of, running the models, because there's a lot of environmental factors that go into it. We can't all say that the lower catch rates are specifically due to, whether it's a toxin or something in the water. But it is a good indication that maybe the sharks aren't in that same area. And these are the same areas that a lot of the reports of spinning sawfish and things like that were coming from. And so sawfish and sharks are very closely related. And so you'd assume that potentially some of the things that would affect sawfish could also be affecting sharks in the same way. So yeah, the number of sharks in the area, the question we don't really know is if the sharks had actually moved out of the area and that's why we weren't catching them, or potentially if some of them, if they're changing their behavior. So if they're having neurological issues, they may not be very enticed to take a piece of, bait on a hook, which is our way of doing a survey. So it's very challenging to say for sure they had completely moved out of the area, but at least we weren't showing the same numbers of them in the survey. We were also seeing for a few individuals very what I would consider to be like bizarre kind of neurological behavior in that a few of the sharks that we had caught, so like juvenile to large juvenile lemon sharks, ones that don't move offshore and they really use these seagrass types of habitat quite a bit. We noticed in a few lemon sharks and also black tips that were in these areas, actually some neurological kind and muscular kind of impairment where when we were working with them, they would almost have like a lockjaw behavior where they're locking up, or their muscles were almost seizing in this like cramping type of behavior, and they were almost unable to release their muscles from these cramps. And so we haven't, in all the surveys we've done, we've never seen that before. And again, this is only a few individuals. We don't necessarily know if it is, potentially related, but if you're seeing neurological issues in other similar related species, this might be, something that's closely related. So as other people, start to figure out what toxins might be the culprit or the likely thing to, kind of hypothesis to go down those routes, then we'll be able to look and run some of these blood parameters, especially for the sharks we've noticed displaying that weird behavior, to see if maybe they are showing any of these signs of those toxins or whatever it might be. So that's where we're at now. We'll be back down there over the summer as well to also to continue some of the survey work too.
Yeah, speaking on the avoidance versus mortality, I know we were the only people to have some tracking gear in the water, and it wasn't much, right? I think 7 sawfish died right on our acoustic receiver that detects animals that are tagged. And during that 3-week period of April, when peak mortalities were happening, we didn't detect a single animal, which I'm interpreting, at least speculating, that there's some avoidance response happening. Good news for tarpon and other things that can move, and bonefish and permit that live in that area, which kind of matches what some people are seeing on the water too, with tarpon in particular. So a little optimism there that not everything's dying. But yeah.
And yeah, so that's what we were working on in April, and then working on this summer as well, is trying to get more of that tracking equipment in the water. Like I said, it's a hard thing because we don't have that pre-data, but hopefully, at least maybe if this event or whatever is causing it starts to wane, at least maybe we can see when animals are starting to move back into some of these environments, if they are in fact avoiding those particular areas right now. But yeah, having that pre-data is sometimes pretty challenging when these things pop up.
Yeah, and going to those sublethal effects too, thinking about running those blood for blood, cortisol levels, and see what the hormonal kind of response is too. Are you planning on doing that as well, or?
Right, so that's what we're going to run a few different blood panels, and so, like I said, we're kind of waiting and trying to pick and choose, because when we're drawing blood, we don't draw a ton of blood from the animals, and so we already have predetermined studies, whether it's reproduction or trophic ecology. So there's not a ton of extra blood left over. And we have to, we're kind of trying to pick and choose to make sure that we're going down the right path. I mean, the other side of it is also, you might not know until you run the blood. You have to try some, but being very selective, I would say, in the routes or in the hypothesis that we're kind of trying to go down. And that's where, being able to work with other people that, are experts in the field and give that guidance, and allowing them to kind of pursue some of these these things first, then maybe that will help us from the shark perspective to know which route to go down.
Indeed, yeah. This is not definitely not a fish kill. The sublethal effects and what that manifests itself long term for these different animals will be something to keep an eye on for sure. And potentially think about what, if nothing reproduces for 2 months or 2 years, or something, because they're so stressed out. What does that mean for the long-term impacts of the fishery? Well, I appreciate it, guys. We're at a half hour now. So I will kind of leave it with a final thought and follow up. This has been a very great collaboration among many people that involves iterative communication and working together, where we, through our collaborative networks, we identify locations for priority for sampling by figuring out which species are the most affected, can inform what do we look for toxins, or what about their ecology that messes up. For Mike, which can inform what Mike does looking for these algae. When Mike finds different algae species, that can go to the toxin testing, and then that all circulates back to improved knowledge and understanding. So as Matt said, waiting to hear from the toxins will inform what he's doing and all that. So this has been great. So a last question for everybody: what on the back end of this, what do we do next? How do we be better? Open-ended. What do we do to make sure this doesn't happen again? If it does end, will it end?
I think it's a great way for us to think about lessons learned and ways forward, what data exists, what data we can, certainly as scientists and, getting grant funding from federal agencies, maybe for small specific projects. But sometimes that data can end up squirreled away a little bit and maybe not out there for everybody to be able to use and harness in these situations. So I think it's an opportunity to maybe get scientific communities together and others, other resource managers and so on, to really take seriously, this idea that we all have and we all really believe in, right, of like data transparency and data sharing. Because I think it's our responsibility to share all of that data so that people can access it, use it as needed, and so that we can really understand these baselines when we need them and in response efforts. That'll be my first thought.
Yeah, I think along those same lines, there are gaps, data gaps, maybe gaps in responses or resources or expertise. And so, what is truly non-existent versus what do we have to go digging for? And it might be networking for people, it might be, digging around for data, like Al said. And I think the other thing, is this a one-off event, or is this something we're going to have to be concerned about? And, can we, if this is something that could happen again, can we get a better idea of what the warning signs might be, or forecasting it? And if we understand the causes or understand, the mechanisms closely enough on how the fish are being affected, can we remedy, can we mitigate at all? But we still need to work to understand what's going on before we can really consider any of that. So it's it's a long process. We're working hard. Al and her team are at it all hours of the day and night, and weekdays, weekends. And so there's a lot of effort, a lot of people involved, as you said, Ross. So, but there's it's going to evolve, and then we'll start getting into more of the how can we better respond to this, and how can we potentially mitigate these type of events if they happen again.
I think something I might just add, if I can, I'm sorry, I'm sure Matt has something as well. And just really thinking about how important it is to have this kind of engagement with not just scientists, right? Like also with people that are on the water every day. Because if it wasn't for the observations of the guides and folks at the Lower Keys Guides Association and others that are on the water, like the commercial fishers and so on, then, we might have found this even later than we did, that it was happening. So I think that's
eyes and ears on the water is really important.
Yeah, 100%. And so I think we really need to give kudos to all of those folks for their efforts and all of this and continued efforts, because they're also helping us sample, right?
Yeah.
And helping to guide like day-to-day knowledge on where things are happening and the changes that are happening. So that sort of thing is just really important to acknowledge, because they're also working hard and trying to maintain their livelihoods out there. So
Yeah, for sure. And yeah, I like that. And even, in the shark research world, the accessibility of people, whether it's in the commercial fishery or recreational fishery, of being able to contribute to that. Because, we all love being out doing research, but in reality, we're only out for a few weeks, maybe a few months if we're lucky per year, whereas they're out most every day that they can be on the water. So being able to include them has been really valuable, even for, tagging studies or whatever it might be. It gives you a lot of really good information. And then from our perspective, I would say, and obviously I'm sure this is to all of you, is kind of resonates about funding, but the value in some really long-term funding that can allow you to not always be reactionary, but have whether it's something like this or whether it's an oil spill or something, having that baseline data, even though it's a shifting based on the baseline we have for 2022 probably isn't accurate to historical, but at least it's somewhat of a baseline before issues like this come up. And so it's really valuable. And I would say for other researchers also being able to sometimes think outside the box of maybe what question they're answering at the time. And so for us, from the Sharklab perspective, we've always approached it as take as much information and data as we can from an animal, as long as it is not adding any undue stress or additional stress to that animal. So if we were catching an animal, say catching a lemon shark for a tagging study, we would pretty much always collect blood from it, even if, as long as the animal was doing well and not being stressed out, even if we didn't necessarily have a designated project for that blood at that moment, because then you have this ability for when an issue like this arises, or when a collaborator reaches out to you and says like, hey, we're noticing something really weird in this region, can we compare it to some blood you maybe have from that region? You already have a bank available for it that you can help provide to other people outside of maybe what your initial goal of your project was. And so being able to really, sometimes have that extra biological samples or whatever it might be can really help other people that when they see something like this.
Yeah, no, I agree. I was just talking to somebody about that. But yeah, for somebody like myself who's been the whole career has been Florida, what this event screams to me, as it does everything in Florida, is like we are a system that's dictated by disturbance and how well our system can absorb or resist those impacts. Hurricanes, those are our big changers. Cold fronts, heat waves, droughts, you name it. Florida's gotten by surviving these things by having healthy ecosystems. And the more we degrade them, the more these disturbances, these events keep insulting the system or lasting longer, having bigger effects. So this tells me that we need to do a really good job of prioritizing things to make our systems resilient to these events. So when they happen again, the system can at least absorb the impact or recover very quickly. So that's improved water quality, doing what we can to keep the habitats healthy, no boats running over seagrass beds. If that may be a stressor that's causing these things to happen, in addition to other stressors, water quality needs to be good. All those things, we need to do a really good job of prioritizing these and of figuring out with science, how do we best prioritize them so that we can be resilient to these events in the future. So with that, I guess I appreciate everybody's time, and we'll check back in when we have something new. And thank you everybody for tuning in.
Thanks, Ross.
thanks so much.
Thanks. Have a good evening.
You too.